Good afternoon, everyone. The webinar will begin shortly. Well, welcome everyone to the fourth in our series of environmental science webinars. Before we begin, I have just a few housekeeping items. I want to remind you that all audience members are muted and your video is turned off. However, we would like this to be a conversation, so please, if you'd like to submit questions, do so by using the Q&A button at the bottom of your screen. So today's talk will address a major human-caused alteration in the Earth's nitrogen cycle that is affecting coastal systems around the globe. Before I begin, you'll see that the MBL students have had a tremendous input into the research that we'll hear about today. And the MBL has had a long tradition of training students from around the world by linking research with education. SES, our Semester in Environmental Sciences program, exemplifies this tradition. For the past 22 years, the program has engaged nearly 400 undergraduates from more than 60 colleges and universities in this intensive hands-on program. Students in the program spend about 20 hours a week in the lab and in the classroom, but they cap the semester by designing and conducting independent research on questions that they are interested in full-time during the last weeks of the semester. Most of the research focuses on ecosystems here on Cape Cod, but many of the topics studied focus on questions being investigated as part of the larger research portfolio of scientists and faculty here at the MBL. During today's webinar, you will hear from a recent alumna of the SES program, Sarah Messenger, who came to us from Trinity College in 2016 and completed her senior thesis based in part on her project studying permeable reactive barriers during the program. Sarah then returned to the MBL after graduating to become a TA in the SES program and is now a research assistant at the Ecosystem Center working on the Toolik Lake LTER. In fact, she just returned from her second semester doing research at the Toolik Lake Field Station on the North Slope of Alaska, where she is responsible for carrying out long-term monitoring and experiments on the lake. And she'll tell you something about that at the end of our session. But now I have the great honor of introducing the other speakers in today's webinar. First, Jim Galloway is a Sidman Pool professor in the Department of Environmental Sciences at the University of Virginia and is a trustee of the MBL. He is a renowned biogeochemist who studies natural and anthropogenic controls on chemical cycles at the watersheds to global scales, and just this year was elected to the National Academy of Sciences. In recent years, he's focused on understanding the beneficial and detrimental effects of reactive nitrogen as it cascades between the atmosphere, terrestrial, and aquatic ecosystems. Jim is a frequent visitor to the MBL, spending sabbatical leaves here and summers at the Ecosystem Center, and has participated in the SES course and mentored many students through that experience. Next, we have Javier Loret, who is a research scientist at the MBL Ecosystem Center, with expertise in understanding how land use change and nutrients flow from land to sea are affecting our coastal ecosystems. He received his doctorate at the University of Murcia, where he studied the role of coastal lagoon benthic ethnic communities in processing nutrients and their contributions to buffering the ecosystems from eutrophication. He has worked on sites ranging from the Mediterranean to Cape Cod. And last but not least, we have Ken Foreman, who has had a long-standing interest in the role of nitrogen in coastal ecosystems. His doctoral work focused on the effect of nitrogen fertilizer additions on the benethnic community of salt marshes. After receiving his PhD from BU's BUMP program, he completed a postdoc recently nearby, and he has been involved in municipal wastewater planning as a member of the Falmouth Planning Board. But very importantly, he has also directed the SES program for the past 20 years. So with that, I'd like to turn the webinar over to Jim.
Thank you very much, Linda. Appreciate the kind of introduction to this seminar on human impacts on the global nitrogen cycle, nitrogen in coastal zones, the good, the bad, the ugly. Right at the beginning, I need to say as a disclaimer that the speaker order has nothing to do with the title of good, bad, and ugly. Now I'm going to share my screen. And we'll start off. So I'm going to share with you what reactive nitrogen is. How have we, as a population, disrupted that cycle? Why do we even care about it? And what can be done about it? And that's where Javier and Ken will come in to focus on the coastal ecosystems. The first one, what is reactive nitrogen? There's two classes of nitrogen in the environment. One, inert nitrogen on your left, the N2 molecule, 78% of the Earth's atmosphere is what we're breathing in a lot right now. And then there's all those things on the right, reactive nitrogen. So what does that mean? Well, N2, very stable, and some processes convert it into things that react with other things. Now there's three types of reactivity. Nitrogen can be biologically reactive as a fertilizer. It can be chemically reactive in the atmosphere, smog, acid rain, stratospheric ozone destruction. And it can be radiatively reactive because nitrous oxide is a greenhouse gas, and that contributes to not only greenhouse gases in the atmosphere, but also stratospheric ozone depletion. It's important to know what are the processes that convert N2 into reactive nitrogen. So the natural processes are shown here by pictures: biological nitrogen fixation in the top two panels, lightning, of course, in the bottom panel. Here is a bit more about each. So we have a special class of microorganisms that can convert N2 into an ammonia compound associated with legumes, primarily in the nodules, and that actually essentially feeds the plant. And then that, of course, feeds the surrounding environment. And then lightning. Lightning strokes lots of energy, and you have N2 and O2 reacting to form NO and NO2. On a global basis, biological nitrogen fixation in the oceans and in the terrestrial land is much more important than lightning. We have some naturally occurring sources of reactive nitrogen. And let me just say something I'll reiterate later on. We have to have nitrogen to live. It's an essential nutrient. Organisms cannot survive without a source of reactive nitrogen. So we have poop. We have guano. The scale here, you can see there's a mountain of guano. There are some people. So this has accumulated over hundreds of years in these islands off the coast, in this case, Peru. And humans have gone there to mine them and use it as a fertilizer. You also have saltpeter, potassium sodium nitrate, that accumulates in arid regions, in this case, Chile. And so these materials were actually taken around the world and taken to places where fertilizer could be made. And of course, the guano factory in Woods Hole was a great example of that. Well, those natural sources worked out fine for a while. But in 1898, this distinguished gentleman here, Sir William Crookes, president of the British Association for the Advancement of Science, said, we're running, the civilized world, that's probably mostly the UK at that time, was running out of reactive nitrogen to grow food. And he challenged the chemists in the academy to solve the problem. Well, chemists came to the rescue. And two of them here, Fritz Haber and Karl Bosch. Fritz discovered the process, invented the process to react N2 and H2, high temperature, catalyst, high pressure, and you make ammonia. Karl Bosch scaled that up to the industrial scale. Both received the Nobel Prize for this in separate years. But the reason they did this was not to feed the world. The reason they did this was because in World War I, the Allied powers blockaded German ships from that source of Chilean nitrates from which they could make munitions. And so Germany, as this says, searched for another way, and that way was the Haber-Bosch process. So we have the natural processes that create reactive nitrogen here, cultivation of legume plants, biological nitrogen fixation, and we have lightning. The human processes are combustion of fossil fuels, coal, fuel oil, gasoline, etc., natural gas. Here we have some coal. This is a Haber-Bosch factory, H2 plus N2. These factories like this are scattered around the world, generally where there's sources of methane, which is the feedstock that produces the high H2 gas. And this world has the capability to feed any number of people on this planet forever, because you need to have N2. There's lots of it in the atmosphere. You need to have H2. We can make it from natural gas. But there's a problem with this, of course, or else we wouldn't be talking to you today. Early on, nature controlled the amount of reactive nitrogen in the atmosphere, in our waters, in our soils. Back in 1850, we had around 1.4 billion people. This is how population has changed with time. This is now the creation rate of reactive nitrogen by fossil fuel combustion. I've put a new axis on here, nitrogen, reactive nitrogen creation, teragrams of nitrogen per year. And a teragram is 1 million metric tons, or 10 to the 12th grams. It's a lot of nitrogen. And so over the course of time, slowly fossil fuel use, of course, has increased, and reactive nitrogen creation has increased from fossil fuel combustion. So natural ecosystems aren't the only ones that use biological nitrogen fixation to get its nitrogen. Cultivated systems, soybeans, for example, use the same thing. Humans plant the soybeans, and so we refer to this as cultivation-induced biological nitrogen fixation. It's caused by humans. Excuse me. And then, of course, we have the Haber-Bosch process. Started out here for World War I purposes primarily, and then really took off as the population of the world started to grow. And you needed to have more nitrogen available to grow food for the world's population. This ends in 2005. Here we are in 2020, and the lines essentially keep on going straight up. How does this compare to what nature does? Here's the total, around 200 teragrams of nitrogen now. Here's the natural range for biological nitrogen fixation on continents. 60 to 70 to 80 teragrams in nature. And so it's a fact that humans make 3 to 4 times as much reactive nitrogen as natural terrestrial systems. I get in arguments with my carbon friends. I say, well, look, carbon's a small perturbation. There's not much impact happening at all. Nitrogen, you have 3 to 4 times as much reactive nitrogen in nature than nature supplies. That's a big problem. To sum that up, here we have this N2 reservoir in the atmosphere. You burn fossil fuels, and all of that reactive nitrogen goes into the environment, directly emitted in the atmosphere. This is reactive nitrogen created by accident, just like CO2 is created by accident. Food production. Here we have reactive nitrogen N2 in the atmosphere, using cultivation-induced biological nitrogen fixation, our Bosch process. We're able to create monocultures of grains to feed us, to create things with feet for us to eat. This is a feedlot in the western part of the United States, and this is not snowy covered mountains. These are burning piles of manure that burned for months. Then, of course, this is being converted to reactive nitrogen on purpose. We need it to create the food to feed the people. This is the Rivas family from North Carolina with a week's supply of food. This is a relative arrow showing how much of all this reactive nitrogen created actually entered a human mouth. And of course, much of what entered the human mouth leaves the human body. And if it's not treated, and I think Ken will address this very specifically, not treated or captured in some way, that's lost to the environment. And this is all the nitrogen used to produce food, but never enters a human mouth. It's called virtual nitrogen, and it's put into the environment by virtue of our creating food. So it's not a stretch to say that most of the reactive nitrogen on a global basis created by people enters the environment. Once it enters the environment, it will produce smog. It can impact forests by soil acidification and impact fresh waters by stream water, lake water acidification, coastal eutrophication, climate change. That's that nitrous oxide, and also ozone, which is formed by NO in the atmosphere. And of course, nitrous oxide contributing to the stratospheric ozone depletion. These are interesting impacts. What's really interesting to me as a biogeochemist is that if you put one atom, one molecule of NO into the atmosphere here from fossil fuel combustion, that can chemically transform and physically transform, and that same atom of nitrogen can contribute to this forest impact, acidification, eutrophication, climate change, ozone hole. It's a cascade. So one element of what one atom of reactive nitrogen created has this multitude of effects. And of course, it's cumulative with time. So we really do have this nitrogen dilemma. We need nitrogen. It's necessary for life. We have to have it. But it has a host of negative impacts on the environment and on people. And so the challenge is to optimize the use of nitrogen while minimizing negative impacts. Now, Javier is going to now take over and talk about this very specific case study of eutrophication, too much nitrogen going into our coastal ecosystems. Thank you very much. Javier, over to you.
Thank you, Jim. Let me see if I can share my screen too. So, well, probably one of the major impacts that this global alteration of the nitrogen cycle has had in the environment actually happens in the ocean. And that is because much of the nitrogen that we put on land, whether it comes from the fertilizers that we're using to grow our food, or from the wastewater from our towns and cities, or from the combustion of fossil fuels, as we were discussing, much of that nitrogen ends up in the ocean, transported by our rivers, the rain, and the groundwater. Nitrogen is the limiting nutrient for primary productivity in the oceans, but when we put it there in excess, it causes a cascade of events that ultimately impact the environment in this coastal zone, and causing a process that we call eutrophication. A good way to give you a sense of the magnitude of the scale of this problem is this map from Selman, which is constantly updated. And each one of these red dots that you see on the map represents a coastal ecosystem that is currently being impacted by excessive nitrogen inputs and suffering this problem that I'm calling coastal eutrophication. There are two things to note here in this map. The first is that there are points everywhere, highlighting the global nature of the problem. And the second one is that the points tend to accumulate in densely populated areas, heavily industrialized areas like the eastern coast of the US, Europe, and the coast of China and southern Japan. The impacts of these eutrophication processes in the ocean are relatively well known and include massive algal blooms like the one on the photo. This is macroalgae, but it can also be phytoplankton blooms, losses of habitat like seagrass loss, It usually comes with lowered oxygen concentrations in the water, which can cause fish kills and shellfish kills, and an overall decrease in the water quality of the coastal zone. I'm going to try to explain how this process of eutrophication works in the coast with these diagrams. What you can see in these drawings is the profile of a shallow coastal ecosystem. In natural conditions, that would be the first drawing on top, concentrations of nitrogen are relatively low in the water, and we usually observe low phytoplankton densities in the water. The waters tend to be clear, and if the systems are shallow enough, they're usually, the bottoms are usually covered by seagrasses. If we add nutrients through our human activities on land, and these nutrients reach the ocean, this nitrogen, what happens is that nitrogen-loving algae, macroalgae, or phytoplankton, sometimes both, grow out of control. And they outcompete seagrasses that usually disappear from the system. If we keep adding nutrients further, what happens is that this overgrowth of algae and phytoplankton overwhelms the system. At one point, big masses of algae will start to die, and the decomposition of that organic matter will consume the oxygen in the water. And that's when we observe fish and shellfish kills in these ultimate phases of eutrophication in the coast. So let me go back to the map, because I'm going to use these 3 hotspots for eutrophication around the world as case studies, just to give you a sense of some of the study areas that I have the opportunity to work on. And I think that illustrate very well this problem of eutrophication on the coast. So I'm going to start in Europe, in the Mediterranean Sea, with the Mar Menor Lagoon, a place that I studied during my PhD a few years ago. And it's a very interesting case. It's one of the biggest lagoons, coastal lagoons in the Mediterranean Sea. And it's also very important in terms of biodiversity. Unfortunately, the land surrounding the lagoon is very densely occupied by intensive agriculture, with huge inputs of nitrogen from fertilizers, and mass tourism that will increase the nitrogen coming from wastewater. The graphs below show, the one on the left, the incredible increase in the area occupied by intensive agriculture that multiplied by 4 in just a few decades in the area. And the graph on the right is just a comparison, just to give you an idea of the seasonality of the population in this area because of mass tourism, comparing the year-round population of 45,000 people living in the area surrounding the lagoon with the almost 500,000 people that come to the area every summer, only during the months of July, August, and September. So all that extra nitrogen entering the system has caused a profound transformation of the ecosystems in the lagoon. What I'm showing in these maps is the evolution of the vegetation covering the bottoms of this particular lagoon in southern Spain. The first map in 1950 shows big patches of seagrasses here in red as the vegetation covering the bottoms of this lagoon, looking something like figure number 1 here. In the 80s, in the early 80s, just a few years after the development of the land started to take place, the bottoms of the lagoon were rapidly colonized by macroalgae, here depicted in green. And sometimes they grew together with seagrasses, and that's the yellow areas that you see here. Just a few decades later, in the 2000s, that's when I was working in the area, most of the bottoms of the lagoon were covered by a dense monospecific bed of the algae, like the one in picture number 2 here. Unfortunately, and after decades of constantly increasing nitrogen loads to the system, the entire ecosystem collapsed in 2016. We observed during the summer a large phytoplankton bloom, like the one in picture number 3, that limited the amount of light reaching the bottoms, which killed all the algae that was growing in the bottom, all the macroalgae that was growing in those bottoms of the lagoon. The decomposition of that organic matter consumed all the oxygen, and millions of fish died as a result and were washed up on the shores of the lagoon. Unfortunately, the system has not recovered since, and all that you see now is like what I'm depicted here in this image, very narrow fringes of algae growing only around the perimeter of the lagoon in the very shallow areas. Another very interesting case happens in the second one of those hotspots that I was highlighting in the global map of eutrophied sites in the Yellow Sea coast of China. The area is experiencing what they call green tides. It's large algal blooms, floating macroalgal blooms of this species, Ulva prolifera. That's the name green tide because it's a green algae. And it looks like this photo in which a ship is sailing across huge masses of algae that go up to the horizon, covering large areas of the Yellow Sea. I got the opportunity to play a little bit with the data just to figure out how these events develop. And what you can see in these images is that the profile of the Yellow Sea coast with China on the left, and the algae will be represented in green and yellows. What you can see here is that these events, these algal blooms, start very close to the coast in the southern part with relatively low densities of algae. But then after only 4 weeks, almost the entire area of the Yellow Sea is covered by very dense masses of floating green algae. It's very difficult to wrap your mind around the scale of one of these events. And so what I did is I overlaid the map of the state of Massachusetts so you can get a sense of scale. And it turned out that the area covered by this bloom was actually twice the size, the entire state of Massachusetts. So you may wonder, well, in order to fuel one of those massive events, you're gonna need a lot of nitrogen, right? So where is that nitrogen coming from? So in order to answer that question, we analyzed the stable isotopes content in the algae. I'm not gonna bore you with the chemistry of the isotopes of these compounds, but what you need to know is that we can use them to figure out if the nitrogen in the algae comes either from wastewater sources, from fertilizers, or from the deposition from the atmosphere. And it turned out that the wastewater was the major source of that nitrogen, with equal contributions from the other two sources. So basically what it means is that the land development, the urbanization and industrialization of the coast of China is responsible for the occurrence and magnitude of these huge algal blooms that they're experiencing every summer since 2007. I'm gonna end in the third of the hotspots that I was highlighting in the global map in the east coast of the US, and particularly on Cape Cod, where we are located. It's a very interesting case because the Cape has experienced and is still experiencing a very rapid urbanization of the land with huge inputs of nitrogen coming from wastewater mostly, and that nitrogen is entering the local estuaries and little ponds and small bays that are around the Cape. And of course, as many other areas in New England and across the world, we have experienced here on the Cape large algal blooms like the one in this photo, losses of seagrass, fish kills, etc., etc. We have pretty much integrated this topic of nitrogen pollution and eutrophication into the SES program. And we use the local estuaries and ponds to learn things about, for example, the connectivity between land and sea, and calculate and measure inputs of nitrogen to estuaries and systems across the Cape. We have studied the impacts, and we continue to study the impacts of nitrogen on food webs, on aquatic food webs. And we also use stable isotopes as tracers to identify the major sources of nitrogen that are polluting these systems on the Cape. And of course, I also had the opportunity to work side by side with many of the students of the SES program in their independent research projects, in projects that ultimately interest them, but that can give also a very interesting dimension to my own research here at MBL. It's always a lot of fun and one of the highlights of my year. We have worked in many topics related to these problems, like the pollution with nitrogen in salt marshes and estuaries here on the Cape, the combined effects of eutrophication and sea level rise, for example. We've also studied the ecology and physiology of the ulva species, the same algae, the same group of algae that causes these green tides, like the one that we've seen in the Yellow Sea in China and other parts of the world, and many other topics across the years. I think I'm gonna stop it here and pass it to Ken, who I think is gonna tell us a little bit more about Cape Cod and about some possible solutions to these problems. Thank you.
Well, thanks, Javier. Let's make sure that this works. So I like to say that in the SES program, we study global problems in a local context. And you've heard so far from Jim and Javier about the global change in the nitrogen cycle and its widespread effects, both bad and good. And I want to try to address how to fix the problem of coastal nutrient pollution. Specifically, how do impaired estuaries and coastal bays recover? We know quite a bit about how nutrient inputs degrade water quality and damage ecosystems, much less about how they recover. And also, what are some of the remediation strategies that can be implemented to cure this problem of coastal nutrient pollution? So there are a number of steps involved in recovery. The first, of course, is we have to reduce the external inputs of nutrients to these systems. But in addition, we need to burn off the accumulated stores of carbon and nitrogen that have accrued in those sediments over many years of eutrophication. And then there is no guarantee that the desired biological community will recolonize. So this may require an active restoration effort. In terms of remediation strategies, the classic one, of course, is when the culprit is wastewater, is the construction of a sanitary sewer and advanced centralized wastewater treatment. But we're also exploring, as part of our research and something we've again integrated into the SES, the use of permeable reactive barriers. And as you will see, they are considered permeable because they allow water to flow through them, but they contain media that reacts with the nitrogen and converts it from reactive end to unreactive end. So that's why they're considered a barrier. So in this slide, you can see an aerial photo of Cape Cod, the coast of Cape Cod, which is fringed by small bays and estuaries, making this an ideal place to study these topics. And many of these bays have restricted tidal exchange and are sensitive to nitrogen pollution and are now considered to have impaired water quality by the Commonwealth of Massachusetts. So there is increasing pressure to reduce nutrient inputs to these ponds. And as these remediation strategies are implemented, this presents a great opportunity for us and our students to investigate how nutrient impacted estuaries recover, as well as explore new strategies for nutrient remediation. What you can see on this figure is the location of the Falmouth Wastewater Treatment Facility. We do have a small wastewater plant in the town of Falmouth. Here's Woods Hole. And this plant treats about 400,000 gallons per day currently of wastewater drawn primarily from Woods Hole, from downtown Falmouth, and most recently from Little Pond. In 2015-16, a sewer collection system was constructed around Little Pond. So I'm going to talk a little bit about that and how we're studying how the pond responds to the removal of that wastewater input. In addition, we've spent a lot of time looking at the impacts of nutrients in Wacoit Bay. This began back in the 1990s with the Land Margin Ecosystem Research Program initiated by Ivan Valiella, and continues to this day. And this is a site where we've located these permeable reactive barriers to test that strategy. So currently on Cape Cod, most of the wastewater that enters these estuaries is derived from single-family homes that release plumes of nitrate-rich or nutrient-rich water into the aquifer, where it's converted to nitrate, carried to the coast, and then emerges along this seepage face in the intertidal zone. And that ultimately degrades water quality. As I mentioned, nobody likes that. It's bad for tourism. So there is a considerable pressure to remediate this nitrogen. How do we do that? How do we convert reactive nitrogen to unreactive nitrogen? Well, the first thing is, when the source of nitrogen is organic N, we need to decompose it to the form, to mineral nitrogen, or the form of ammonia. So here are, you can see my phytoplankton, perhaps zooplankton fecal pellets, or as Jim mentioned, poop, being converted to this mineral form. Once it's in the environment, there are microbes that can exploit that high energy ammonia compound to grow. They actually are carbon fixers. They're called nitrifiers. And in the process, if oxygen is present, they do this creating nitrate. And that process is called nitrification. So the process of removing nitrogen, or converting it from reactive to unreactive form, we call denitrification. We begin with nitrate, so that's why we need this nitrification step. And then if we have anoxic conditions, which are the result of the decomposition of high energy compounds like sugars and cellulose, there are microbes that use that high energy compound and the nitrate as an oxidant in the process, releasing CO2, converting that organic carbon to CO2, and converting the nitrate back to this unreactive, stable, inert, as Jim mentioned, N2 gas. And that's exactly what happens in an advanced centralized wastewater treatment plant. So every year, we visit the Falmouth Wastewater Treatment Facility. You can see students in the upper right-hand side of this slide looking into the pump room of that facility. Effluent or influent comes into that plant, passes through a series of screens and chambers and settling tanks, and has at that point a concentration of about 20 to 40 milligrams of nitrogen per liter. It then flows into these sequencing batch reactors you see in the lower portion of the slide. These are large tanks that alternate between aerobic and anaerobic conditions. So they have a vigorous aeration phase during which organic matter is decomposed, and ammonia that's released is converted to nitrate. And then they enter a quiescent phase when the tank, the liquids in the tank go anoxic. All the oxygen gets utilized. And the next thing that happens is the nitrate is consumed through this process of denitrification that I just explained. And the net result is that the treated effluent emerges with a concentration of somewhere between 2 and 5 milligrams nitrogen per liter. So that's what's happening in your local wastewater treatment plant. That's what's happening in the Falmouth wastewater treatment plant. And as I mentioned, back in 2015-16, the watershed surrounding the Little Pond estuary here sewers were constructed within that watershed to hook up about 1,400 homes to this wastewater treatment facility. In the process, we expect to remove about 80% of the nitrogen load that's currently impairing water quality in Little Pond. So in anticipation of that, what's really happening here from the point of view of an ecologist is a major ecosystem level experiment. We built a network of a dozen groundwater monitoring, multi-level groundwater monitoring wells, in which we can collect about 46 discrete samples to look at how water quality changes in the groundwater as it's flowing into the pond, as the effect of this sewering takes place. And here's what we have found so far. Taking advantage of the SES program, we visit this system every year. We sample this well network. We've done this in July 2016. So here on the bottom graph, you can see number of homes hooked up to the sewer system through time. Sewage construction was completed in 2016, and homes gradually began to hook up. And over the course of the next 3 to 4 years, nearly 1,400 homes have been hooked up, and we've sampled prior to any homes being hooked up, July 2016, in November 2017, July 2018, November 18, and November 19. And hopefully before the end of the year, we'll collect another set of samples. Along the way, if you take the average of all the well data that we have at each of those sampling dates, you can see there's been a general decline in the concentration of dissolved inorganic nitrogen that's in that groundwater. And I'll remind you, dissolved inorganic nitrogen is merely the sum of the nitrate and the ammonia present. So the amount of ammonia present is shown by this lower line here, and it's stayed relatively constant. And most of this decline is due to the removal of nitrate in the groundwater. We were not sure we were going to see a pattern like this because groundwater flows fairly slowly through the aquifer, about a foot per day. And so there was no guarantee that we would see such a rapid response in the reduction of nutrients in groundwater after the hookup to the sewer. And that was one of the reasons we undertook this project. So a questions that are left to address is, will treating just a wastewater input be sufficient to cure the eutrophication problems? After all, that is not the sole source of nitrogen input. We also, to these systems, we have fertilizers that are applied to people's lawns. We have acid deposition. And then, as I mentioned, there's this multi-year legacy of contaminated groundwater. So it might take quite a while for that to clear out. It does look promising in the Little Pond area. And finally, it's quite costly to build sewers. The construction of the sewer collection system around Little Pond alone cost $40 million. And the estimate for the Cape as a whole to do this, to help address the problem throughout the Cape, is billions of dollars. Is there another way to remediate end pollution? Why do we have so much nitrate in the groundwater? Well, the reason we do not seem to have denitrification occurring naturally is that the sandy aquifer of Cape Cod does not have enough carbon to support high rates of microbial metabolism that would lead to the anoxic conditions in the aquifer necessary to denitrify. In the meantime, there's plenty of oxygen in the surface soils and in the unsaturated zone above the water table, so that nitrogen leaching from these septic systems is nitrified on its way to the water table. And once it's in the form of nitrate, it's quite mobile, moves with the groundwater to the shore. So we had the idea. We're not, of course, the first originators of this idea. It actually originated with Will Robertson at the University of Waterloo in Ontario. But we had the idea to implement a test of the use of a permeable reactive barrier composed basically of wood chips to supply the carbon source that along the coast to intercept nitrate-contaminated groundwater and see whether or not this might be an effective strategy for remediating that nitrate pollution. If effective, it would capture all sources of groundwater borne nitrogen borne to the estuary. If you placed it right at the shore, it would become effective almost immediately. The discharge is at the site of origin, and a PRB would be cheaper to build and operate passively. It has no operating or maintenance costs. So in 2005, with funding from the SICE program of NOAA, we installed 2 test barriers at seacoast shores along the Charles River and at the head of Waquoit Bay. This is what it looked like when we were installing these barriers. One is about 20 meters long, the other about 12 meters long. And here's how we went about sampling those. We installed a network of permanent wells, both up gradient from the barrier, across the barrier, and down gradient from the barrier. So these sample at multiple depths. And then we also were able to basically march along the seepage face in the intertidal zone with a portable well point sampler that collects groundwater from various depths and sample that groundwater just as it was about to emerge into the estuary. Here's what some of the resulting data looks like. So the top graph here, I show you a concentration of nitrate in micromoles in the Charles River system. This is the location of the barrier. We have horizontal distance here in meters, location of the barrier between about 0 and 15 meters. And you can see nitrate levels were quite comparatively low, 100 to maybe 10 or so micromoles compared to region outside the barrier where concentrations range from 400 to 700 micromolar. Similarly in Waquoit Bay, and this is a log scale going from 0.1 to about 1,000 micromoles, where there was no barrier, concentrations range from 10 up to 150 or so micromoles. Where the barrier is located, concentrations were drawn down below 1 micromole. And in fact, you can see this if you look along the seepage zone. The barrier here is located, this is the head of Waquoit Bay, here where the vegetation has been disturbed. We've covered it up with sand and it's now revegetated. But groundwater is flowing from up gradient across the barrier, emerging here in the seepage zone in the intertidal. And here at sort of low tide, you can see that we get an algal bloom every spring where that fresh water that's nutrient rich is emerging into the estuary. And where the barrier water is emerging, no algae grows, presumably because there are no nutrients. And you can also look at this in terms of in cross section. So this is a vertical slice through the beach. Here we have depth in meters versus relative distance in meters horizontally. This is the grade along the beach. Depth 0 is approximately sea level. And this is some of the very first data that was collected by SES student for her research project in 2005, Sabrina Moreau. Each of these vertical lines and nodes represents a sampling point in a permanent well. The orange box represents the location of the PRB. And the warmer the colors, the higher the concentration of nitrate. Peak levels here in the middle of this plume were about 150 micromoles. Once they impinge upon this barrier, the nitrate plume is removed. Now, we also had a student interested in the genes responsible for nitrate reduction. Nitrate denitrification, dissimilatory denitrification involves a number of biochemical steps, one of which, conversion of nitrite to NO, is catalyzed by the NIR gene. And Colin Knaus decided to try to look at whether the NIR-S gene was more prevalent in the region of the barrier versus up gradient from the barriers. So he collected groundwater from each of these nodes again, and then looked at a copy number compared to nanograms of total DNA present. You can see that there's a tremendous hotspot in the region up of the barrier itself and just slightly down gradient from the barrier. And finally, I want to mention that the disappearance of nitrate alone is not definitive evidence that it's all being converted to N2. So we've done controlled experiments, again, with the help of students. Here's Allison Tucker from Mount Holyoke College, where we dug up wood chips from the PRB. We put them in these columns. We upflowed groundwater. We saw the disappearance of nitrate. and the accompanying appearance of N2 using membrane inlet mass spectrometry, which allowed us to precisely measure how much excess N2 was present. Presumably, this has all been converted from that nitrate. And there's quite a good match showing quantitative conversion of nitrate to N2 as this groundwater flows upward. The last question that I often get asked is, what is the lifetime of the wood chip media? And again, we've done column studies, reactor column studies, now using wood chips that are 14 years old and looking at their performance in removing nitrate compared to reactor columns filled with new wood chips. And here's some of that data. See here again, nitrate concentrations on the vertical axes, residence time in the column on the horizontal axis, new wood chips shown by the orange line and the warmer symbols consume all of the nitrate loaded. Here we're loading on quite high concentrations, 12 milligrams per liter, within about 6.5 hours. In that same interval, these 14-year-old wood chips, although less effective, still managed to consume about 40% of the nitrate, and within about 21 hours, most of that nitrate has been consumed. So they do seem to have a long life. This might be a very viable solution to, at least in some locations, to remediate nutrient pollution. And I just want to thank all of the students and collaborators who've helped with this project, including a number of funding agencies, SciSeed, Sea Grant, and NSF. So with us today is one of these students who has worked with us on the PRBs and also is continuing to work with us at the Ecosystem Center, Sarah Messenger, and I'm going to hand the talk over to her.
Thanks, Ken. So I'm just gonna share my slides here. Yeah, so as Ken and Linda have mentioned, I came to Woods Hole as an SES student in 2016 and then worked on a project with these permeable reactive barriers with Ken. These are just a couple of pictures of me in the field at Wacoit Bay. And I extended that project into a summer in Woods Hole and then also my senior thesis at Trinity College, where I used molecular tools to explore the role of sulfur-based denitrification in the barriers. And then I came back to the Ecosystem Center as a TA for the SES class of 2018. And I stuck around to work with Jim and Javier on the MBL's nitrogen footprint. So where the interventions that Ken just talked about address reactive nitrogen that's already in the environment, this nitrogen footprint project looked at reducing the amount of nitrogen we release to the environment in the first place. And I'll move through this really quickly, but we calculated the amount of reactive nitrogen that's released as a result of the MBL's activities, which you can see.
Sarah, we're not seeing your slides.
Oh no, sorry.
No problem.
Let's see. What is going on? Oh, how about now?
Perfect. Now we're seeing them.
Oh, okay.
Thanks.
I'm so sorry. Thanks for bringing that up.
Absolutely.
Yeah. Okay. Oh, so here I am in the field. That's Wacoit Bay. This is what I was talking about with the nitrogen footprint project. So this is on the left. This is the amount of reactive nitrogen released to the environment as the result of MBL's activities. And then once we had that, we looked at different ways that we could reduce our emissions. We focused mostly on dining related scenarios because, as Jim mentioned, that's a primary way that humans are releasing excess nitrogen to the environment. And we also looked at some fossil fuel utilities related scenarios and found that the MBL could reduce its nitrogen footprint by 8% if it combined all of these modeled strategies at the same time. So that's just another, tool in the toolkit of reducing the negative impacts of reactive nitrogen. I've kind of transitioned from that project now, and as Linda mentioned, I'm a research assistant on the Arctic Long-Term Ecological Research Study. So I've been lucky enough to spend the last 2 summers in Northern Alaska doing field work, and these These are just some pictures of me there, studying nitrogen among many other things. But it all started out with those permeable reactive barriers and SES, and you never really know where you're going to end up. But I really have Ken and the SES program to thank for giving me the tools and the opportunities to pursue this kind of research and really opening up a whole new world for that. And with that, I think I'm going to hand it back to Ken for some Q&A.
Well, thanks, Sarah. There is time for a couple of questions, and I see one that is probably directed to me, so I'm going to answer that one. First, it says your end measurements at the PRBs are very shallow. I thought much of the N2 laden groundwater flowed much deeper. How do you stop that? So I want to be clear that the experiments we were doing were a test of concept. They were not intended to be a final step in remediation, but locating the barrier close to the shore, you know, the lens of groundwater, fresh groundwater, becomes quite narrow there and emerges up through, you know, it's underlain by salt water. Now, that's not true everywhere. There are some of these systems where perhaps the fresh water flows under the estuary, but I think most of the evidence is that the freshwater flows are deflected upward, and the bulk of that fresh water emerges through that seepage zone. So the closer you get to the shore, the shallower you need to put your barrier. On the other hand, if you put it close to the shore, you have to worry about sea level rise, so there are other issues. There is currently a project underway in Falmouth in the upland area using PRBs that's intended more as an actual remediation strategy. And I like to think that they're building some of their effort on the work we've done to demonstrate the potential use of PRBs. And they're using a different carbon source. They're using emulsified vegetable oil, which can be injected to depth. So that project is injecting this carbon source 50 feet down into the aquifer, where based on earlier studies, we know most of the nitrate plume is located. So that addresses that question. Let's see what else we have. So seems like agricultural practices, this is probably for Jim. Jim, if you're there, please.
I'm here. Thank you.
Seems like agricultural practices for processing for food production are incredibly inefficient. If only a small fraction of the reactant eventually is eaten, are there ways to decrease the wasted reactive nitrogen that is generated by agriculture?
Yeah, it's a great question. And there's three, you can approach it from three ends, so to speak. First, you can make the nitrogen use efficiency of crops and animals more efficient by a variety of techniques. And there's been great progress on that. Secondly, you can decrease on the food consumption end or the food serving end, you can decrease food waste. In this country, the numbers estimate anywhere from a third to a half of food put out in retail, grocery stores, restaurants, is wasted, thrown away. It's very expensive waste. Or third, and this is probably the easiest for a person to do, in addition to controlling food waste, and that's only eat the protein that you need. People in the United States on average eat 40% more protein per day than the USDA recommends. And so it doesn't make any difference whether the protein's animal or vegetable, but by decreasing your protein consumption to what's recommended, you can have a tremendous impact on nitrogen recovery in this country. Thank you.
Okay, well, before you go away, I guess another related question really is, are you seeing any changes in institutional policies as a result of, you know, in terms of reduced end use, as a result of the end footprint work?
Was the question, are institutions changing their behavior?
Yes. Is it effective?
Okay.
Have you had an impact?
We have. In fact, I'm very happy to boast about the University of Virginia, where the nitrogen footprint tool originated. It was published by an undergraduate, Allison Leach, from her senior thesis of 2009, more formally published in 2012. In 2013, the University of Virginia Board of Visitors, the governing body, set a finite goal that by the year 2025, it was going to decrease its nitrogen footprint by 25% relative to 2020 to 2010. And that was in line with the carbon goal. More recently, last December, the UVA Board of Visitors said, well, we're going to make it a little tougher. By 2030, you have to reduce the nitrogen and carbon footprints by 30%. And UVA has just finished up a 5-year project of which MBL was part, where we went out to other institutions. And now there's hundreds, over 600 universities and colleges in the United States who are using a combined carbon nitrogen footprint tool that's run out of the University of New Hampshire. So we're very proud of this study and very appreciative of all the partners involved, including MBL.
Great. Well, thanks. So we've run a little longer than some of our earlier seminars. So I think it's now 1 o'clock, and it's probably time for us to close. I wish we had more time for questions. But I do want to remind everyone that on December 8th, Joe Volino and Emil Ruff will be presenting the next in this series, that is Breathing Without Air, Microbial Superheroes of Our Backyard. We might even hear about a few denitrifiers in that process. So I thank everyone for attending, and I thank all the presenters for participating. Take care.